the anatomy of the neurons. What exactly are neurons? Neurons are the excitable cells of the human nervous system. Earlier, there was a belief that the number of neurons is comparatively less than the number of neuroglial cells. In simple, neuroglial cells or supporting cells outnumber neurons. But recent studies have proved that this theory is actually wrong. In human beings, the number of neurons is almost equivalent to the number of neuroglial cells. However, this ratio is reversed in case of thalamus. Thalamus is one region in the human body or human nervous system where the neuroglial cells to neurons ratio is as high as 17:1, right? This is regarding a brief introduction about the neurons. What exactly are the parts of neuron? A typical multipolar neuron. We will talk about the classification a little bit later, but for now, let's talk about what are the parts of a neuron and what is the structure of a neuron. A neuron typically contains two important parts. One is called as the cell body and the other one is called as neurites. The cell body is known by various names. The cell body of a neuron is also called as perikaryon or soma, whereas the processes of the neuron are called as neurites. The neurites can be of two types. What are the two types? One is dendrite and another one is axon. First, let's talk about the cell body. The shape of the cell body varies with the type of the neuron that we speak. This contains any other cell organelles compared to any other somatic cell. So, it contains a prominent nucleus and a nucleolus. The prominence of the nucleolus will depend on the activity of the neuron. Apart from this, it contains cell organelles like mitochondria, endoplasmic reticulum studded with good number of ribosomes, Golgi apparatus, lysosomes, everything. One of the important component of cell body of a neuron is its endoplasmic reticulum with the ribosomes. In simple, the rough endoplasmic reticulum. The rough endoplasmic reticulum of a typical neuron is modified to form something called as Nissl substance. This Nissl substance is involved in the synthesis of neurotransmitter. The importance of Nissl substance is also in helping to differentiate between dendrite and an axon. So, when we talk about these two processes, we will discuss further. This is in brief regarding the cell body of a neuron. Now, coming to the neurites. Neurites are the extensions or the processes that arise from the cell body. These are dendrites and axons. What do we mean by a dendrite? Remember, dendrite and axon are not defined by their length, but by their direction of carrying of impulses. If a neurite or a process carries the impulse towards the cell body, you call it as a dendrite. If process carries the impulse away from the cell body, you call it as axon. That's it. Okay, it is not dependent on length, it is not dependent on myelination, it is purely on the direction of the transmission of impulse. Right? Then, now if you want to differentiate between a dendrite and an axon histologically, how will you do it? Take a section of the dendrite and take a section of the axon and observe it under microscope. One of the most important striking differences between axon and dendrite is presence of Nissl substance in dendrites and absence of the same in axon. Right? This will help you in differentiating between the two types of processes histologically. Right? So, this is in brief regarding axon and dendrites. Right?